Hi everyone. This PowerPoint is used for a review or for students who were not in class the day we did the lab. Right now we're going to be setting up for the animal meiosis lab. For the animal meiosis lab, you need to work with the partner. I will give you either a pink egg cell or a blue sperm cell. You'll also receive an animal chromosome chart. And on this chart, we'll have 16 chromosomes for your fictitious animal. So this animal doesn't exist. It only has 16 chromosomes. Humans have 46. So this is not a human. You'll need to cut the chart in half. If you do not have a partner, you will be a female, and I will give you whatever you need to complete the lab when it comes time for it. So right now, cut the animal chromosome chart in half. You need some scissors. Brings us to our next step. If you have a male on your animal chromosome chart, you will circle the sex as a XY. If you have the female being the egg, you will circle the XX. What you're representing now is you are a germ cell, and if you are a male germ cell, then you are going to be located in the testes of this animal, and you are going to eventually make 4 sperm from the process of meiosis. If you're a female animal cell, germ cell, you're going to be located in the ovaries, and after the process of meiosis, you will then make 4 egg cells. The next step, I'd like you to cut out all 16 chromosomes and have them at your desk. So pause this play so you cut the chromosomes out and you're ready for the next step. After you're done cutting, I would like you to set up your homologous pairs of chromosomes like a karyotype. So just like a geneticist would, I would like you to arrange the first pair of chromosomes, and then the second pair and the third pair. Let's put those on top, and then we'll have pairs 4 through 7 in the middle, and the last row will have the 6 chromosomes. I also want you to fold the chromosomes lengthwise, so once only one side is showing, remember we're still setting up the simulation. We're pretending right now that the other side does not exist. Okay. After you have completed the karyotype, I'd like you to take a look at your animal chromosome chart. You can see that that is my chromosome chart in the background. I would like you to select a genotype for all of the 7 traits that are given. I have selected the first one for you. So for the trait or gene for coat color, everybody will have the genotype big B, little b, which is heterozygous for black fur. You may choose the genotypes you want from genes 2 through 7. When you are done with that, you want to write on each of the homologous pairs, you want to give each chromosome one of the genes. So, for instance, on chromosome 2, I gave a big K to one chromosome and a little k to the other chromosome. This is my genotype. One gene came from the mother, one from the father. You are only writing on the front side of those chromosomes. Remember, we're pretending like the back of the chromosome does not exist. When you're finished copying all the genes, you should still have your chromosomes arranged in pairs like a karyotype would appear. Are we ready for the simulation? So let's just review meiosis. Meiosis starts off with the germ cell or the parent cell, and there are 2 cell divisions. Before cell division, there's an interphase where the DNA replicates before the cell is getting ready to divide. Then we undergo the first cell division, which is meiosis 1, which produces 2 daughter cells. After meiosis 1, both of these cells then enter meiosis 2. These two cells split, making 4 daughter cells as the result. So during interphase, just like in mitosis, the cell is growing, but the cell does one very important thing, getting ready for the cell to divide. And that is that the DNA replicates. So we can simulate DNA replication by unfolding the chromosomes, and on the other half, which is we now have doubled chromosomes, on the other half, go ahead and write that exact same gene. So if on the left-hand side of the chromosomes, you can see the picture, I had a big B. I would go ahead, unfold the chromosome, and write a big B to model that that DNA chromatid has replicated. And now I have 2 exact DNA strands. The pink dot in the middle represents the centromere. So I have 2 doubled chromosomes in this picture, which is called a tetrad of a homologous pair of chromosomes. So we have a tetrad of a homologous pair of chromosomes. So do that, show me DNA replication for each and every chromosome that you have in your genome. At the end of interphase, after the cell does some more growing, we now go into the first step of meiosis 1. So the first step in meiosis 1, we have prophase. Now we can't model one very important step in prophase 1, which is crossing over with these pieces of paper, but we can lift each homologous chromosome up and make them touch and visualize that they are exchanging genes. So you never do get a gene or chromosome handed down directly from your mother or father. So again, one thing that we can't simulate with this lab is crossing over, which ensures more that the offspring even are more genetically diverse than what would have happened if it wasn't for crossing over. So in metaphase 1, the homologous pairs of chromosomes, they're going to line up in pairs, so that if you're using a desk, the top of your desk would have pair 1, pair 2, all the way down to, used to have pair 7, followed by the sex chromosomes. So the homologous chromosomes line up in the equator or the middle of the cell. During anaphase 1, we're going to pretend that our fingers are spindle, and they're going to attach to the centromeres of each doubled chromosome, and they're going to pull each doubled chromosome to one end of your desk, which is going to represent the opposite ends of the cell or poles of the cell. After anaphase 1, we now have 2 separate cells, as you can see. So we're done with meiosis 1. Cytokinesis has occurred, the cytoplasm has doubled, some of the organelles have doubled, and then there is no interphase after this. It directly goes to prophase 2. So we could say in telophase 1, the nucleus would have reformed, but then in prophase 2, the nucleus disintegrates again, and we're getting ready for the second stage of meiosis, which is meiosis 2. So in order to prepare for metaphase 2 and anaphase 2 of meiosis 2, what I need you to do now is cut the doubled chromatids down right down their centromere and line them up. Remember, we still have 2 separate cells, so we're treating these as we have 2 separate cells on our desk. So once you cut those, again, keep them kind of together. So we're simulating metaphase 2, and we're going to come up on anaphase 2. So once you've done that, then you can simulate anaphase 2. You can do 1 cell at a time. So you will pull the sister chromatids apart in anaphase 2, and you will do that for the other cell that's at your desk. And eventually you will end up with 4 rows of individual chromosomes, chromatids. And what we're going to do, since this is telophase 2, we're going to combine those chromosomes together to show that we have 4 cells as a result of meiosis. So you can see in the picture that somebody is taking those lines of chromosomes that they separated and making 4 piles of cells. So congratulations, you have just made some gametes. You made 4 gametes. If you're a female, you've made 4 eggs. If you're a male, you made 4 sperm. Just a quick fact, if you are a female, only those 3 of those eggs will die off and only one becomes the good egg. If you're a male, all 4 of those sperm become viable, that they have an opportunity to fertilize an egg. But right now, what I would like you to do is to, whether you made 4 sperm or 4 eggs, at random you are going to take 3 cells, and you're going to put those cells in the garbage can, and you're going to have 1 cell left out on your desk. What you're going to do with the chromosomes in the cell is to place them in your sex cell. So place those chromosomes in your sex cell. If you have the egg at your desk, you're going to place the 8 chromosomes in your sex cell. If you have the male sperm, you're going to place those 8 chromosomes in the sperm. One thing to note, again, meiosis was the process of reducing the number of chromosomes by half. So we started off with 16, and you can count that you only have 8 inside of your gametes. Which brings us to the next step of this process, which is fertilization, the fun part. So if you were sitting with a partner, it's the sperm that will have to travel to the egg. So we're going to have the sperm travel over to the egg, and you may dump your genetic information to fertilize the egg. And once you've done this, you can congratulate yourself because you have just made a baby. So you have a fertilized egg, which is called a zygote, and this is how your animal that you've created will start off life, as a single cell. Once you reach this part, you will then align to see the, the new combinations of genes that your offspring has that's different from yours. You will receive a birth certificate. You may name your animal. You may write the parents' names. If there was only one person working, you can ask me and I can provide you with the other parents' genetic information. Once you do have both parents' genetic information, arrange the chromosomes again in their homologous pairs, and go ahead and circle the genotype of your offspring on your certificate of birth. Once you complete that, then you are almost finished. You can show me your certificate of birth and I will provide you with the next assignment. That would be drawing what your animal looks like and answering some conclusion questions.